To get right into it, there are two questions that motivate the research in my lab, and the first one I'll explain by showing you this figure. We all know that a handful of soil or a pinch of feces has more microorganisms in it than there are humans on the planet, which makes it especially surprising that almost everything, I would argue, that we know about microorganisms has been learned by cultivating them as pure species in the lab, yet almost everything they do in the real world is in the context of a complex multi-species community. And so the first question we have is, which many of you have as well, is how microorganisms interact with each other and with larger organisms like us. The second question has to do with these molecules, natural products. And those of you in the room who are physicians or who may just have known these from their reputation will have heard more about these molecules in how they're used in the clinic to treat disease. What I'm going to talk about today is different from that. It is to ask the question of why would a microorganism go to the trouble of making a molecule this complex? The way I'll explain the details of that question is to show you the set of genes that are necessary and sufficient for the production of daptomycin. These genes had been discovered previously. We sequenced the genome of the organism that produces it. This set of genes occupies about 2% of the chromosome of that organism. It's an enormous amount of the genome to devote to a single molecule. And maybe even more surprisingly than that, the in aggregate set of proteins that is required to make daptomycin is larger than the ribosome. And yet it's there just to make a single molecule, right? So an extraordinary investment of carbon and ATP in just one molecule. So the second question really is, what are the natural roles of these molecules that would make it worthwhile for an organism to produce them? And I'll try to convince you that these two questions have, at least in part, a common answer, which is that some, not all, but some of these natural products are there to mediate interactions among microorganisms and then between microbes and their host, for the ones that are host-associated. The thing that my lab adds to the old way of discovering natural products, which is still, you'd be surprised to see how often this still happens, is that we've introduced the tools of genomics and genetics and bioinformatics to the equation. It does two things for us. One is that it allows us to find molecules that we otherwise wouldn't have been able to find. And the one that's more interesting to me is that it gives us a new window into the biology of the organisms that produce these molecules and maybe new ways of understanding why they make them. I should tell you at this point, and this is an important one, that the set of genes that are necessary and sufficient for the production of a molecule like this is a relative of the antibiotic vancomycin, are almost always physically clustered in the genome of the microbe that produces them. They're sitting right next to each other. They're contiguous. And that underlies a lot of the computational work that we've done, which I'll get to in just a moment. I'll also add, apropos of the virome discussion yesterday, that these are not, in my experience, I've never seen one carried by a virus, but they are almost always transposable elements or mobile elements. They often are the entire mobile element. The fact that they're transferred around horizontally has interesting ecological consequences that I probably won't get into today. Let me give you a quick vignette that explains how we got interested in this kind of work. This goes back to the time when I was at Harvard, and there was a very bright postdoc in Roberto Coulter's lab named Paul Straight, and Paul was interested in the interaction between two different organisms in the soil, Bacillus subtilis and Streptomyces silicolor, that you might find in any pinch of soil. And what he did was an interesting genetic screen where he transposon mutagenized the bacillus and then plated it on a lawn of Streptomyces. And what he was looking for were bacillus mutants where their appearance didn't change, but the appearance of the surrounding Streptomyces did change, which would have indicated that he had found a gene involved in an interspecies interaction. Found, in fact, many mutants like that. They all clustered in one gene cluster in Bacillus subtilis. This is the laboratory workhorse gram-positive organism that had been studied for many decades. But this cluster of genes, again, about 2% of the genome, nothing was known about it. That's where I became involved in the story. And this is the time where we were working out bioinformatic tools that allowed us to take a look at this cluster of genes. These are the biggest genes in the Bacillus genome. They encode a set of proteins that act like a molecular assembly line to synthesize a complex small molecule. And we were working out tools that allowed us to predict the structure of the small molecule that would be building up along the length of the assembly line and even the final structure of the product. And so working in collaboration with a postdoc from John Clardy's lab, we were able to figure out that these genes encode this exotic-looking molecule, Bacillaene. This is an unfinished story. Still not clear why interrupting Bacillaene biosynthesis leads to the precocious upregulation of a red pigment by Streptomyces color, and that red is, in fact, a different diffusible small molecule. But it is clear that this is some kind of two-way, small molecule-mediated conversation between these two organisms, and knowing the nature of the small molecule, the structure of it, that is the key factor on the bacillus side, opens the door to being able to tease apart the molecular mechanisms at play in this conversation. Okay? So I'm going to talk a little bit about connecting genes to the molecules they encode and vice versa. What I'll say is that whether you start by figuring out a molecule that you're interested in or a set of genes that are of interest for one reason or another, the thing that we do and enjoy doing a lot is being able to computationally connect genes to the molecules they encode and vice versa. We do it at first computationally, and then we use genetics and analytical chemistry to solidify the connection. And what I'll argue is that doing that is going to teach us a lot about the organisms that produce natural products, which is almost all microorganisms. For a moment, I'm going to take a different entry point to this little triangle, and I'm going to say something which might sound outlandish, but follow me for just a moment, which is that what if we started right here, and we just went through all the bacterial genomes in the database and asked ourselves, can we find all the sets of genes that are probably involved in making a small molecule? If we could do that, and even more than that, we could make a prediction about what those molecules might be. I'm going to suggest that that gives us a different way to learn something about the biology of microorganisms, and so give me just a moment to try to convince you of that. I'll tell you two reasons why it makes sense to start with the genes. The first is that there are lots of genomes that have been sequenced and also metagenomes, and the tools that we've developed are quite good at going through them and finding sets of genes that encode a small molecule. So it is possible to do it, and somebody made a comment earlier about how one of the major bottlenecks now is really analysis of that data. That's the same problem we've run into, and we think that where there's a problem, there's enormous opportunity, and this is one of those places. Second reason to do it is exemplified by this organism named Saccharopolyspora erythraea. This is the industrial producer of erythromycin. So an organism like this will generally be fermented on, 90,000 liter scale in an enormous tank. Much is known about organisms like this, and yet when its genome was sequenced and the other genomes of similar organisms were sequenced, a big surprise emerged, which is that here I'm showing you some of the 35 or so sets of genes that appear to encode a natural product. This one up here in blue encodes erythromycin, which I'm sure you'll agree is a complex small molecule, and many of you know it's a pretty potent antibiotic. The small molecule products of the rest of these are unknown. So this means that we probably don't need to go to the corners of the earth to find new molecules. Maybe we should just go to our backyard or our freezer collection and find a way of tickling these organisms so that they turn on the production of molecules that they probably don't normally make under the artificial condition of cultivation in the lab. So I'm going to tell you a little bit about the computational work we're doing and where it's going. The challenge has been, can we look through a genome sequence and pick out genes that are involved in making a natural product, something that, like many other challenges that are well addressed by machine learning, can be done by an expert relatively easily but slowly. So we've done this, and we have a training set of about 750 known biosynthetic gene clusters with known small molecule products, and we've devised a very simple machine learning algorithm that has recovered for us an enormous set of biosynthetic gene clusters that we think is quite interesting. One of the first things that we noticed about this set of gene clusters, which about 4 years ago really changed the experimental direction of my lab. 3/4 of my lab is experimental, and we used to study soil microorganisms, and now we study the human microbiota. And the reason is that many of these, of the most interesting biosynthetic gene clusters that we found, were not in exotic soil and marine microorganisms. They were in gut and skin and oral bacteria that I had never cultivated before, but now we grow many of them and we enjoy it. I'm not going to tell you as much about the evolutionary implications of what we found, although they're quite interesting. would you, for example, believe me if I told you that this molecule, meridamycin, is more closely related to platyanolide, meaning the genes that encode meridamycin are more closely related to the genes that encode platyanolide than they are to the genes that encode rapamycin or FK520? You wouldn't have guessed that from its structure, would you? Interesting implications for how you would engineer these systems to make new molecules, which is, suffice it to say, a topic of current interest. And I'm also not going to talk much about our efforts to computationally predict the structures of the small molecule products of these gene clusters, although it's a very important process that I think for us generates the best hypotheses that we have to work with. Instead, what I'm going to do is just to tell you a little bit about the experimental work that we're doing, and I'm going to give you 3 vignettes that each of which touch on something that's come up either yesterday or today in one way or another. So we'll see whether you agree with me that there is promise in looking at diffusible small molecules from the microbiota. I'm going to, maybe I'll just say for the sake of ecology that this is not a new question. When I give this talk to my natural products colleagues, I have to do this in order to make them feel comfortable with it, but I say many of you over the last 10 or 15 years, the most interesting work that I've been reading has not been looking at free-living soil microorganisms and looking at microorganisms that are associated with strange sea creatures, or insects, or plants, or worms, you name it. It's hard to find a multicellular organism that doesn't live in close association with a microbe, you know that, but in the natural products world, it's hard to find one that doesn't live in close association with a microbe that's already been shown to make a complex natural product that is important for the biology of the pair. So it really shouldn't be a surprise to anyone that the suggestion that humans might also be involved in this kind of a conversation. Let me acknowledge the people who have done this work who are in the upper left-hand corner, and I'll be happy to take questions if there's time.
Thank you. That's very exciting work you're presenting. Questions? Mark?
Mike, that's really terrific work. Congratulations. I wonder with the natural products that you're discussing, for example, the antibiotics that the Propionibacterium make or the lactobacillus make, do you know anything about the regulation, what's turning them on and off? And when they're induced, what's the level that we're seeing? Are we talking about nanograms, femtograms?
So both good questions. The answer is that it really differs from strain to strain and from molecule to molecule. You can find cases where the molecule is produced under normal laboratory conditions, but that's probably less common than finding cases where it's off under normal laboratory conditions. We find cases where people do this in old-fashioned brothology kind of ways, where you try a bunch of different culture media and elicitors, and some condition will elicit the production of a molecule. But this is it's one of the most active areas of research in the natural products community because it's been such a thorn in the side of groups trying to characterize molecules like these. So we know very little about the regulation. Everybody hypothesizes that there's some cue from either a competitor or the host that's turning them on. If you haven't found a condition under which the molecule is induced dramatically, then it's probably that you're not looking at the right condition, because there are enough cases known where people have looked hard enough and found levels of these molecules that just go through the roof that it would be it would probably be wrong for us to assume that some of them are just turned on a tiny bit. That's unlikely.
Any other questions? I would make one comment. You probably know this, but you mentioned the peptide in the sponges. The sponges do have these primitive immune systems and reject grafts with cells that are like natural killer cells. So they may, I don't know if this is a signaling molecule for the sponge immune system.
That's a great comment. We've had that thought. I don't know how to study it in the sponge context, but I'm guessing the alpha-galactosyl ceramide from the sponge is a super potent inducer of human natural killer T cells, and I'm guessing the same is true for some cell type in the sponge. So I completely agree.
Yeah, I can't remember. There was a person at UCLA when I was there who studied the sponge immune system, and they don't do much, but they do reject grafts. It's quite interesting. Yeah.
I want to cede a moment to Margaret. Were you going to make a comment about sponges? Because I saw your hand go up, and I know you could be.
Yeah. Actually, no, Michael, that was great. I mean, what I was going to say was, it seems to me that where there's a mother lode is in the hypotheticals. And so we know so little about how microbes interact with hosts and how hosts interact with microbes. And I was in a meeting recently, and somebody got up and had done a microarray, and they said, oh, we didn't find any genes that suggested symbiosis. And I said, did you get regulation in the hypotheticals? Oh, yeah. But of course, nobody knows what those are. So this is a great adventure, it seems to me.
David, quick comment.
So, Michael, maybe in the discussion, just like combinations, thinking combinatorial, combinations of genes, pathways, et cetera, maybe the true activities of these things are only seen when they are together in their own combinations as well, right?
Absolutely. So one of the great mysteries, and I'm glad you asked the question, of antibiotics, since it's known from work from people like Jerry Wright, that a soil microorganism might be resistant to 15 different classes, naturally resistant to 15 different classes of antibiotics, and that some of these guys make, conservatively 8 or 9 different antibiotics, some of them at the same time. We have one organism that makes 3 different ribosome-binding antibiotics that all bind at different sites in the ribosome. The question is, what's in it for them? They wouldn't still be making the antibiotics if there weren't something in it for them, since we know these elements get lost very quickly over evolutionary time. And so it could be that acting in the context of a combination gets you something that acting as a single agent doesn't get you. And I actually think that has important implications for the future of antibiotics.
Okay, great. And we do, although I know Mike may have to leave before the discussion, is that right? Or yeah. Okay, but we do have time for further discussion later.